It all started in 1885, when Hans Adolf Eduard Driesch shook two sea urchins' embryos apart, separating the cells. Two individual sea urchins grew from the original set of cells, thereby creating the world's first demonstration of artificial embryo twinning. In 1902, Hans Spemann underwent the same process with two salamander eggs. However, instead of shaking the cells apart, he detached the cells from one another with a strand of baby hair. 50 years later, two scientists named Robert Briggs and Thomas King successfully transferred a nucleus from a fertilized frog egg to an egg without a nucleus. The egg went on to grow to a tadpole. This process was significant because it exhibited that cells could undergo nuclear transfer and still be feasible. Then, in 1996, another breakthrough occurred. Using somatic cells, or cells in the body other than reproductive cells, scientists Ian Wilmot and Keith Campbell were able to clone the famous Dolly the sheep. They did this by implanting the nucleus from a somatic cell into an egg cell. This development was especially momentous because it showed that organisms could be cloned by implementing cells other than ones used for reproduction. Another major discovery occurred in 1997 when Angelica Schneike, Keith Campbell, and Ian Wilmot transferred a genetically engineered nucleus into an egg cell that had its nucleus removed. The result was the birth of Polly the sheep, a counterpart to Dolly. Before long, scientists underwent the same processes used to create Dolly and Polly, and were successfully able to clone a myriad of species, including mice, goats, and cows. As scientists continued to experiment, they came across another innovation in 2007. Using stem cells from the rhesus monkey, Shakrat Metallopov and his associates were able to transfer the nucleus of a stem cell into an egg cell. The cell began to divide and further develop, generating the first case of cloned embryonic stem cells. More recently, in 2013, the same scientists were able to undergo the same process but with human stem cells. Although they didn't allow the cells to mature into an embryonic state, it is still a possible measure scientists could carry out in the future.
There are many present-day uses for cloning. Most of the uses for cloning are young and in need of research, but that is what scientists have been up to. But even though this process is still in the works, it is important to talk about types of cloning that are used. There are three main types: gene cloning, reproductive cloning, and therapeutic cloning. Gene cloning is a type of cloning that makes copies of genes or certain segments of DNA. The main purpose of this type of cloning is to compare and study different versions of DNA sequences and genes for information on the nucleotide sequence of a gene. Reproductive cloning is the cloning of full animals. There are a few ways that this cloning is possible. One way is that a scientist can take a skin cell of an animal they want to clone and put it into an empty egg. When the egg starts to grow, it is then placed into the womb of an animal. Another way is that scientists fuse an entire somatic cell with an empty egg before placing it in the womb. Therapeutic cloning is cloning that is used for medical purposes. This includes producing embryonic stem cells for experiments that are aimed at creating tissues to replace injured or diseased tissues. These could include organs.
The largest medicinal contribution of cloning comes in the form of embryonic stem cells. These cells are created by taking adult DNA and injecting it into a fertilized egg that doesn't contain a nucleus. Once the embryo has formed the cells needed, they are harvested and the embryo is destroyed. The stem cells can then be used to create new heart muscle, brain tissue, bone tissue, and essentially any cell in the body. This is an extremely huge advancement within the medical field because it allows for patients to receive organs made from cells with their DNA so that their body doesn't reject it. It also applies to reparation of cells. It could be used for spinal cord injuries or heart defects and has even already been used to restore someone's eyesight. However, it is not without its downsides. It comes with massive ethical flashbacks due to the fact that these stem cells are derived from human embryos. In an effort to steer away from using human embryos to create stem cells, not only because of ethics, but also because of the scarcity of human eggs, a different method has been introduced. This new procedure consists of taking a sample of skin cells and using proteins, converting them into induced pluripotent stem cells. These cells are, in essence, adult cells that are genetically reprogrammed to resemble embryonic stem cells.
With technology that we have today, the idea of creating all sorts of life by ourselves is not seen as impossible. With this idea of genetic cloning and recreating stem cells, there are many applications that we can use on other species. The medicinal applications are limitless, from keeping us safe from many diseases to creating whole organs in just a matter of days. One distant application that genetic cloning can be used for is creating life itself. Throughout the creation and testing of genetic mutations, scientists have created life from almost nothing. One of the many ways that this process can be used in the future is in our livestock. In the US, pigs and cows are the top leading animals that provide for our food. Dr. Ken White and his group at Utah University are working to produce animals not to be consumed, but to be breeding animals. By using the genes and cells from a carcass of these animals, they can create a superior breed that is able to pass on its genes to more of its offspring. One crazy theory includes drug vaccine producing cows. To break it down more easily, they would get cows that produce a high amount of milk. They would then input a gene into their DNA, which is basically code for the type of drug or vaccine that they would want to use, with one cow passing it on to its offspring, creating an entire herd of cows with drugs in their milk. Creating life is exciting enough, but what about bringing back animals from the dead? Now, I don't mean a bunch of theory. It could be possible for humans to recreate dinosaurs and other ancient extinct species, but what they would need is a well-preserved DNA sample, which is pretty much impossible to find given they existed thousands of years ago. Cloning also brings the possibility to preserve more species, but it doesn't help the problem that wiped out the animals in the first place. In this case, cloning could be more of a way to conserve the species rather than to release them into the wild just to have them die off again. But the real question that always pops up is, could we clone humans? The answer is simple: Star Wars. That means yes. The real problem with cloning humans isn't the scientific side, but the social side. It is seen as highly immoral, and there are many laws keeping scientists from researching further. Some are all for the idea of creating clones, seeing it as a way to help with infertility, to help advance society, and even the horrific idea of having a farm of clones to harvest organs. Nevertheless, these possibilities won't come up for a while, and perhaps not at all until we have a good idea of the risks involved.